This is an overview analysis of Dr. Bauer's research article, FOXP2 Mediates Sex Differences in Ultrasonic Vocalization by Rat Pups and Directs Order of Maternal Retrieval by Colleen Valentine and Amanda Ward. Here we'll discuss some more background information about the FOXP2 gene. There are many sex differences seen in nature. However, this research article focuses on the auditory and vocal differences. One of the main examples of this are mating calls. This can be seen with birds throughout nature and how males have a different song to attract the females and vice versa. However, the mating calls are one of the main described differences in nature, but this article also focuses on the parent and offspring relationships, which is less talked about. One of the main suspected causes of this vocal difference between the sexes is the FOXP2 gene. The FOXP2 gene is a well-conserved gene that is seen across a wide range of species. Typically in nature, the males have a higher abundance of the gene than females, but the opposite is true for humans. Also for humans, the gene can be traced back to chimpanzees and Neanderthals, which also represents how it's across a wide range of species. The FOXP2 gene also functions as a transcription factor, where it regulates both the repression and expression of genes associated with brain patterning, specifically with neurite growth, dendritic branching, and axonal morphology. Mutations in the FOXP2 gene can lead to language and speech impediments such as stuttering or apraxia of speech when the brain gets damaged. Relating back to the birds example, a lack of FOXP2 causes them to not be able to imitate other birds and will vary their songs. Now this experiment focuses on rats and the FOXP2 gene specifically. Rodents are special in that they communicate via ultrasonic vocalizations, or USVs. And when newborn pups are isolated from their mothers, which are also called the dams in this experiment, they can emit distress calls in USV form. There are two main purposes for this experiment. First is to determine whether there are identifiable quantitative sex differences in rat pup ultrasonic distress calls, and if so, what would mediate this? And the second is to determine whether the dam or the mother rat perceives the sex difference and alters her behavior accordingly. As for materials and methods, there were two main samples used. We used newborn pups from the Sprague Dolly rats litter, and then we also obtained human tissue from boys and girls post-mortem, an average of about 4 to 5 years. And all of these human tissue samples were taken from the left hemisphere and the Brodmann's area. Various tests were used to identify the FOXP2 gene within the tissues, such as Western blot analysis, immunohistochemistry, and siRNA treatments. And then PCR was also used to amplify these to better study the gene. And then also ultrasonic vocalization recordings were used to test if there were differences that the human ear could hear between the sexes of the pups.
We reviewed the results of the experiment. Male and female rat pups were killed after 4 days since birth, and Western plots were performed on the cerebellum, amygdala, cortex, thalamus, hypothalamus, hippocampus, and preoptic area to detect FOXP2 protein. T-tests were performed to compare males and females. Males had significantly higher basal levels of FOXP2 protein than females in the cerebellum, amygdala, cortex, and thalamus. The cerebellum had a gene expression level of 4.24, the amygdala at 2.54, the cortex at 3.23, and the thalamus at 3.914. There was no sex difference in the FOXP2 levels in the hypothalamus or the POA. There was also no FOXP2 protein detected in the hippocampus for either sex. In addition, there was no sex difference in the amount of protein levels for the closely related FOXP1 in any of the brain regions. The sex difference is due to the generalized reduction in FOXP2 protein in each cell for females compared with males, at least it's suggested. Identifiable sex differences in rat pup ultrasonic distress calls were recorded and quantified. The ultrasonic calls of individual 4-day-old pups that were departed from the dam for 5 minutes. Isolated pups made hundreds of calls during the separation. Male pups averaged almost twice as many as the female pups. Average duration of the individual calls ranged from 60 to 80 milliseconds and did not vary between males and females. A 2x2 ANOVA test indicated significant main effects of the sex. Male pups produced more total vocalizations than females. The impact of treatment with an siRNA was directed against FOXP2 mRNA on pup USVs. A two-way ANOVA indicated a significant interaction between treatment and sex. The control males produced 603 total vocalizations compared with 350 produced by control females. The siRNA-treated males produced 466 total vocalizations, which was fewer than the control males. siRNA-treated females produced more total vocalizations, and for siRNA-treated males to produce fewer total vocalizations was not a hypothesized outcome. Control males vocalized a lower frequency than control females, and the treatment with the siRNA was reversed. Bowers was interested to learn in mice the sex differences in the FOXP2 protein levels in the brain and how they are higher in males than females. Sex differences are apparent only in areas of the brain associated with higher cognitive functioning, but not reproduction like the POA and hypothalamus. Different levels of the FOXP2 in males and females are controlled in the developing brain. Separation in distress calls from neonates demonstrates to parents the need to be retrieved and modulated as shown by suppression of calling in response to predator detection or pharmacological treatments. Selective breeding for high versus low rates of separation induced USVs by rat pups reveals genetic underpinning that co-evolved with the effective behaviors. Determining whether the sex difference is genetically driven or a consequence of differential hormonal exposure during perinatal sensitive period for establishing sex differences in the brain is still being determined. Isolated from dam control, males had increased number of total vocalizations, a lower frequency, and lower amplitude than control females. Males treated with FOXP2 siRNA had female-like USVs, reduced total number of calls, and higher frequency. Phenotype of females with reduced FOXP2 was very similar to control males. It had an increase in the total number of vocalization, lower frequency, and lower amplitude. Opposite effects of FOXP2 siRNA in females is unknown, but may reflect a rebound and subsequent overshoot in FOXP2 expression due to inhibition starting from lower baseline, reaching a critical threshold for reactive expression. Sex-specific basis in maternal care is presented in anogenital grooming, preferred on male pups versus females. This could be due to testosterone-dependent olfactory cues from males. Our selected species, like mice, have shown a manipulation of sex ratios in response to environmental conditions and produce large numbers of rapidly maturing offspring. Olfaction ruled out by anesthetized pups, which do not call, and was not retrieved by the dam. The data also looked at gender differences in humans involving speech and language. There is a clear gender difference in articulation with speech, ranging from acoustic parameters of frequency and pitch to speed of vocal production. Girls produced communication skills by first using pointing gestures earlier than boys. There isn't a language advantage in girls in humans. Girls produce their first words and first sentences at a younger age than a greater variety of sentence types and early communication than boys of the same age. The FOXP2 gene was analyzed in 4-year-old boys and girls. Boys had a lower expression rate, and the FOXP2 levels are associated with more communicative sex. There is data that suggests male rat pups might be more effective communicators using USV, whereas in humans the linguistic advantage of girls is well known. As for critiques of the article, the layout of the paper is not a formal research paper necessarily, because the data is seen in materials and methods section when it should only be depicted in the results section. In addition, the methods should be explained a little bit more because we had to research some of the tests. As for experiment critique, how it applies to the real world is that FOXP2 gene is present in humans, but has not been experimented or researched greatly in the past. We discovered that the FOXP2 gene is more present in girls than boys at around the age of 4, which is similar to the age of pups in comparison. We also learned that age 4 is a significant development age for language. As for future experiments, other animals could be tested to demonstrate a well-rounded study, such as other species instead of the Sprague dolly rats. We could also test if similar distress calls are made between the species. Another test could be to test different ages of the pups and the mothers and see if the UVSs are different. Another experiment that should be run is to test if the FOXP2 gene is present also in the right hemisphere, for humans specifically, as the tissue samples were only taken from the left hemisphere. Lastly, we could test pups and their USV calls to different mothers and see if vocalization occurs.